This video is about enzymes. So as you probably remember, enzymes are made of protein, meaning they're made of amino acids, and they get their shape from the way that those amino acids fold based on how the R groups of the amino acids interact. Enzymes have an active site. That's where the substrate binds. They act on that substrate by catalyzing a reaction. So that could be taking two substrates and connecting them to be one piece, or it could take one piece and break it into two pieces. Different enzymes do different things. Also, some enzymes have a spot on them where an allosteric or non-competitive inhibitor could bind, that when that binds would change the shape of the active site so the substrate can no longer bind. There are also competitive inhibitors. Competitive inhibitors are the same shape as the substrate, and so they can bind in the active site and block the substrate from entering the active site. Enzymes' major role is that they decrease the activation energy of a reaction. So enzymes basically speed up reactions that would have happened anyway. And this is a graph that we looked at when we were learning about this, where the x-axis is the progress of the reaction, and this axis, the y-axis, is the energy. I actually wrote reactants twice, I'm noticing. This is the energy of the reactants. This is the energy of the products. And without an enzyme, this reaction would take about this much activation energy to get over so we can get to the products. But with the enzyme, this line here, we need less activation energy to get to the point where we can move to the products. So it just makes it easier for a reaction to happen. The environment can impact how enzymes function. So for example, the pH can impact how well enzymes function, and the way that the pH can influence enzyme function is by disrupting its shape. So we know that the R groups on amino acids, this is amino acids in a chain, can have charges on them, like positive or negative charges, and those charges can cause those R groups to attract and cause the protein to fold in a certain way. Now, if the pH changes, that influences the charges of the R groups, and so interrupts this bonding, and so those R groups aren't attracted anymore, so the shape of the protein changes. Temperature and salinity and inhibitors can all also affect how enzymes function. Temperatures can help increase the rate that a substrate is interacting with the active site to speed up the reaction, but it can also cause denaturing of protein as well by interrupting the way that those R groups are interacting with each other. Salinity also can influence how protein is folded. And inhibitors, as we saw in this picture, can function in a couple different ways. They can be allosteric and change the shape of the active site, or competitive and just get in the way of the active site. Those are all things that would be in the environment of the enzyme. So you might see a graph like this, where we can see what the best pH or the best temperature is for an enzyme. So for example, in this graph, we can see amount of product produced. This could also be like the rate of the reaction or something like that. And we see a peak here at pH of 5-ish. So this particular enzyme happens to function best at pH 5. Different enzymes have different pHs that they function best at. Like the enzymes in our stomach that break up food function better at a lower pH, a more acidic pH. Also, this is a graph of temperature. So this enzyme happens to function best at like 75 degrees Celsius. That's pretty darn warm. So that could be an enzyme that's like in a hot spring or something. Our body temperature is around 37 degrees Celsius, just so you know. I also want to remind you of the enzyme lab that we did. So we looked at an enzyme called catalase that we got from potatoes and yeast. And catalase breaks down hydrogen peroxide into water and oxygen gas. So the way that we measured how quickly that hydrogen peroxide was being broken down is we put our catalase, our yeast slurry or potato slurry, on little filter discs. And as the catalase produced oxygen gas by breaking down the hydrogen peroxide, those oxygen gas bubbles collected on the filter paper and caused the filter paper to rise. So you might remember that you tested out different things. You chose to either test the salinity or the temperature or what else? The amount of hydrogen peroxide. You tested different things and collected your data. And then we presented to each other in class and tried to figure out how those different things in the environment can affect how enzymes function, and we were able to kind of see the effects. Another thing that I want to talk about is another type of graph that you might see about enzymes. So you might see a graph where it's time on the x-axis, product on the y-axis, and it might ask you how a line would change if something was added or removed from an experiment. So in this, the brown line that I'm tracing right now with my marker, that's my baseline or my control. If we added more substrate, then the line would look more like the orange line. Notice that the slope is the same, so the time that it takes the product to be created is the same, but what is different is where it levels out. So there's more product than in my control, which makes sense because the more substrate I put in, the more product I can make. If I added more enzyme, that's my green line. So you can see that the slope here is steeper. So product is being made faster. And that makes sense because if there's more enzymes to break down the substrate or to build up the substrate, the reaction's gonna happen faster. You'll notice that it plateaus at the same place though. We didn't change the amount of substrate. It just is faster to get to that amount of product. If we added a competitive inhibitor, you would see a lower slope or a decreased slope in the amount of time that it takes product to be formed, because the inhibitor would be preventing the enzyme from doing its job, and so it would take longer for that product to accumulate because it would be taking longer for the enzyme to have the substrate in its active site to make the substrate into the product. But again, you'll notice that it reaches the same peak because we didn't change the amount of substrate in that case. We were just changing how effective the enzyme was by adding an inhibitor.